Okay, well, the good thing about my talk is the last one, so we're almost done. I'm doing something slightly different because I'm going to talk a little bit more about our line instead of one specific project. My title, my Plan A title, was using genomic techniques to look at diversity of non-model small mammals from tropical Asia. But this is very similar to what other members of my group have been talking about for the last few years. So I changed it, but not in the program. Wow, it looks terrible. Genomic tools for studies of ancient DNA. So we are using genomic tools in what we call ancient DNA, which encompasses a variety of materials, and basically doing phylogenetic analysis for a few different reasons. So when you think of ancient DNA, primarily think of paleontological material or subfossils, for example, from caves. One of the places where we get samples is this really beautiful cave called Ardales, south of here. And they dig the bones out of the bottom of the cave. But we can also work on DNA from archaeological material. In some cases, in most cases, the question is what organism did the modified artifact come from? And then also from historic material from museums. These gets back to South Asia are some of the tree squirrels that we're working on from tropical South Asia. So for the more ancient DNA from paleontological materials, we can address both population level questions, but are also doing a lot of species level questions involving species which are extinct. And one recent example that we participated in was the phylogeny of this scimitar-toothed cat in relationship to the saber-toothed cats and the other extant large cats. And also the divergence within this species, which was very widespread from Europe to North America. And so we had looked at it before in North America, and what this added was a sample from Europe, which there are extremely few of. So this was very interesting to see how very, very close the North Sea, that's the European one, is extremely closely related to the North American one and had very much less diversity than other large cats. So for the archaeological questions, we're often asked what species some modified archaeological sample came from. So it could be a bone that has been turned into a tool and no longer has the characters that would allow you to morphologically identify it. Or in one case that we recently did, in a renovation of the Alhambra, they found some old Muslim documents which were illuminated, which means they had pictures drawn on them, which is extremely rare. And based on the morphology, they thought that it was horse skins, which would also be extremely unusual given the religious context. And so we took several small samples across this very large document to identify what species skin had been used in making parchment. So they all turned out to be cow. So yeah, the morphology was wrong, and it was the more culturally appropriate material. And here's another example of, this is manton for a virgin from Seville, and we worked on the DNA of these, all these little heads and stuff are made out of ivory. And so we actually did a genetic analysis of the ivory during the renovation of this modern culturally important item. And then what connects any of this to tropical South Asia is a lot of our questions about the evolution of this fauna go beyond the samples that we can collect ourselves. And so we complement a lot of the samples we get in our fieldwork with material from museums. And this is a ground squirrel. And so this is the one species that we actually work with and trap alive. And all of its relatives we had to sample in museums because this species lives here in northern Borneo, and all of its relatives are up here. And it turned out that those weren't its relatives, and it belonged in a different genus. And so this tells us not only about the evolution and history of this one species, but about the biogeography of the region, and that these deeper levels of genera, not just species, are divided along these biogeographic routes that are established. So things are more compatible with the rules than they were thought. And another example of this is the squirrels of the same subfamily, which are Specios in this region, which is called Sundaland. But a small number of them have crossed the biogeographic barrier called Wallace's Line, which goes right here, and are in Sulawesi, which is this sort of K-shaped island. And so we looked to see how the squirrels in this area called Sulawesi are related to the ones on the other side of this biogeographic line called Wallace's Line. And so this work on the Dromomies is with mitochondrial DNA, which is easier to work with historic materials. So in the beginning I was going to say, the thing about ancient DNA, one of the problems with working on it is that it's broken into very small pieces. But that's okay with a lot of genomics tools because the first step is to break it. So you just skip that step, it's already done, and put it into your library and start sequencing, right? And so the last example then is with these Sulawesi squirrels. And so we usually start with mitochondrial DNA because it's easiest. And using genomics tools, we can get a lot more data and look at whole genomes or larger genes instead of just small fragments. But when we look at the mitochondria, what I'd like to bring your attention to is that Procellarius here in green, they're here and then up here. So that genus looks polyphyletic. It's broken by these other two genera. And this is the mitochondria. But for these, we also worked with UCEs, ultra-conserved elements, which is an enrichment kind of a system of markers that are extremely conserved across tetrapods. And this was actually a test to see if they could be useful on this small scale as they were developed to look at questions on a very much deeper level. But we were able, with historic material, to enrich for these UCEs and get sufficient variability to look at the relationship between these closely related species and these closely related genera. And with the nuclear markers, the phylogeny fixes itself and our genera all come back together. And this highlights the importance of using lots of markers for phylogenies. So basically, we're using genomic tools with degraded materials and making phylogenies for a number of reasons, from identifying species to looking at the evolution of specific groups and of biogeographic regions. So thank you.